Good morning, good afternoon, good evening, colleagues and friends from the 4 IOIS regional federations. My name is Rosana Pelayo, Principal Investigator at the Mexican Institute for Social Security and at your service in the IOIS Education Committee. Thank you all for joining this IOIS webinar series, Immunology Without Borders. This program, in partnership with Immunopedia, provides the opportunity to discuss hot topics in immunology research, to unite the global immunological community, and to respond to unexpected pandemics like the one we are facing. We wish to thank Fluidine for supporting the production of this topical webinar and promoting international cooperation. Today, it is my great pleasure to introduce our invited lecturer, Dr. Andrea Cossarizza, an extraordinary scientist, always open to share knowledge and promote collective work in such a dramatic time for humanity. Dr. Cossarizza is full professor of pathology and immunology at the University of Modena and Reggio Emilia, located in Modena, northern Italy. He is also director of the School of Specialization in Clinical Pathology and Clinical Biochemistry and vice dean of the School of Medicine. Andrea holds a master's degree in medicine and surgery from the University of Padua and a PhD in oncology from the Universities of Bologna and Modena, with a specialization in clinical pathology and immunohematology from the University of Modena and Reggio Emilia. His scientific activity is mostly focused on immune responses and predictive markers in patients under immunotherapy, immune alterations during HIV infection, autoimmune disorders, and COVID-19, of course. And he has generated a fascinating body of work on immunological changes in aging and inflammation. He has become a highly productive opinion leader in advanced cytometry of disease and is past president of ISAC. Of note, he ran a race from immunology in the fight against disease in Cancun Beach last 2018. In this webinar, relevant findings on clinical aspects of mild and severe SARS-CoV-2 infection in pregnant women or elderly will be discussed. Moreover, we will learn about therapy and vaccine responses. So Andrea, thank you for taking the time to be with us. We welcome you to this IOIS program. And before I leave you with the audience, just a reminder to attendees, please make your questions in the Q&A chat. I will take them to Andrea after his presentation. If you have any sounds problem, please make sure your browser is not on mute. Welcome, Andrea.
Well, thank you, Rosanna, for this very nice presentation. Apparently, you show someone who is a big guy, but I'm not like that. I'm just a normal person who is doing immunology on different topics. So I will present you some of our most recent data on what happens during COVID-19 disease and SARS-CoV-2 infection. And I will focus on two particular aspects. I mean, those that you can find in women during pregnancy, or those that you can find in old people who have, of course, a completely different immune system in comparison with young one. Before starting, let me show my disclosures. I had grants from different companies, banks, association, and private citizens to study COVID-19. And I'm very proud to tell that most of the grants arrived immediately after the first patients in March 2020. And thanks to the generosity of a number of people and a number of companies, including BioBiotech, Thermo Fisher, Beckman Coulter, Beckman Dickinson, Agilent, Biored, and Fluidine, we were able to start immediately to study patients with COVID-19 in the very first days after the pandemic. So what is happening is that we know well that the lungs I would like to go ahead with the slide, but apparently it's not so easy. I cannot go ahead. Okay. Well, we know that inflammation is the first and maybe most important driver of the immune response. Inflammation is playing a major role during the pathogenesis of COVID-19. And we know after more than 1 year that the control of inflammation by drugs that are able to block some parts or some molecules is crucial for the survival of the patient. We have learned that different conditions exist in which after an infection with SARS-CoV-2, inflammation is triggered by a number of different molecules, and we know that if you control such molecules, you can have a good clinical result. Some examples of the control of inflammations are those coming from pregnant women, and pregnancy is a typical condition in which the immune system is skewed toward Th2. On the other side, there is aging, which is skewing towards Th1, and during aging process, there is the phenomenon of inflammaging that we have described more than 20 years ago, which is a status of some clinical chronic inflammation that continues the production of inflammatory molecules and can provoke serious damages to the organism, especially during an infection. So we think that understanding the basis of the endogenous regulation of the phenomenon of inflammation could be very important to design novel forms of therapeutic strategies, or to better design or to better use old and new vaccines. And we know that the lung is the first organ which is infected by the virus, and cells like those present in the bronchi, like epithelial cells, as well as type 1 and type 2 alveolar pneumocytes and capillary endothelial cells are infected by the virus. So what happens is that signaling molecules released by infected cells and alveolar macrophages, in addition to those recruited by T lymphocytes, monocytes, and neutrophils, can start the inflammation process. Then, at the later stage of the progression, a pulmonary edema exists that fills the alveolar space. And this is followed by the formation of membranes, hyaline membranes, that cause what we call early phase acute respiratory distress syndrome. And obviously a number of anomalous coagulation phenomena can exist that result in the formation of microthrombi and all the subsequent thrombotic sequelae. We know that the immune response is important, of course, it's crucial, and we have a huge number of molecules and cells that participate to the immune response to SARS-CoV-2. And in fact, after the epithelium is infected by the virus, the replicated virus can cause cell lysis and direct damage to the epithelium. But the epithelial cells present viral antigens to CD8 cells that with the perforin, granzyme, and so on, including activating the activity of NK cells, this immune system can provoke the killing, can kill infected cells and induce apoptosis, trying to stop the infection. However, if this is the good part of the story, the bad part of the story that several mechanisms can be triggered that increase the damage and on the one side or on the other side provoke a limited response to the virus. And we know that we can have a massive recruitment and activation of granulocytes that go to the lung and provoke a massive damage to lung cells. We can have dendritic cells with a lack of production of interferon. We can have, on the other side, a massive production of pro-inflammatory molecules. We can have an increased local and generalized inflammation with the release of DAMPs and the stimulation of other inflammatory molecules that provoke further damages. Finally, we have to mention that the production of cross-reactive antibodies that recognize different coronavirus, antibodies that are produced against other coronavirus but cross-react with SARS-CoV-2 can be pathogenic and can, in a way, reduce the ability of the immune system to cope with this particular infection. So, in the last year, we have performed a number of studies on SARS-CoV-2, and we have published the first paper on March 18, and this was a sort of position paper in which we asked ourselves and to all the scientists which are the challenges for global health that we have to cope with in terms of the response to such a new virus. Then a few weeks later, we took the responsibility to write a position paper in which we wrote how you can handle safely handle blood specimen from patients with COVID-19. And then we also did a number of other studies that were involving T cells, B cells, monocytes, granulocytes, and genetic mechanism of illness, clinical illness, disorders, and international collaboration. Then we published a study in Lancet Respiratory Medicine that reviewed a number of information. We had another study in the Lancet Rheumatology that I will show you in a moment. And then we performed a collaboration with a group of Pierre Christopher and Ton Schumacher in Holland, in the Netherlands, to understand then characterize a specific CD8 T cell response and the immune dominant features of lymphocyte responding to the virus. Then I will start from what we found. The first data that we could publish were those related to the activation, senescence, exhaustion, and differentiation of T cells. So what came out immediately after the first patients was that the T cells were a little bit confused, because among this population of lymphocytes, you could find cells that were activated, exhausted, senescent, or that show an alteration in the differentiation. And we found, indeed, as you can see here on the bottom, a number of cells that were, in a way, more activated, but also more senescent and more exhausted. And this was one of the first information that the scientific community had concerning the T cell response. Then we performed a little bit a more complicated analysis with an unsupervised methodologies in flow cytometry. These are staining with 18 color flow cytometry, in which we use UMAP, which is a uniform manifold approximation and projection. Is a system to see in one single graphic here on the upper left part of the slide what happens in a complex population, and on the right you can see the flow sum analysis, and you can see that different populations were changed in patients with COVID-19 and pneumonia induced by COVID-19, and you can see alterations in the amount of naive Tregs, in the effector memory cells, sentinel memory, and other populations. And these are CD4 T cells. What happens in other populations like CD8? If I could go ahead with the slides, which I cannot. I'm sorry, I lost the cursor. Okay, thank you. Similar data were found also among CD8 T cells, in which the populations that changed were very, very more impressive. And in fact, using the same approach of the UMAP and flow sum, we found a number of cells that were altered, in particular naive T cells, as well as activated cells, naive CD38 positive cells, and those who show markers of exhaustion. So to make a long story short, what came out was that a number of parameters related to T cells were completely altered in the early phase of the response to the virus. Then we took a look to mass regulator genes and chemokine receptors, and we found that those that were able to help inflammation had a significant change in patients with COVID-19 pneumonia. And in particular, what we found was that a population of cells that you can see on the right part of the screen, those expressing CD161 and CCR6, were significantly decreased both among CD4 or among CD8 T cells. And this is very important because those cells here are able to go to the lung and produce IL-17 that in turn recruits granulocytes and other inflammatory cells, and in turn increases the pulmonary damage. So again, we did not find particular changes in mass regulator genes like TBET, GATA3, and so on, but we found consistent changes in the expression of chemokine receptors, indicating that some cells with the ability to produce inflammatory molecules were able to go to the lung and do their job. And indeed, what came out when we studied the production of inflammatory cytokines was that cells from patients were able, both CD4 and CD8 cells, were able to produce more IL-17, and this was something very important for us because it put a little brick in the wall of pathogenesis. And what was interesting was also that if we studied the polyfunctionality of these cells and you can see the case in the upper right part of the slide, was that a number of cells were able to produce more than one molecule in the same moment, and in particular we found a consistent increase of cells able to produce IL-2 and IL-17 together. And IL-17, we know well, is very important because it's the molecule that is able to recruit neutrophils to the lung, and in any place, of course, but if your Th17 cells are going to the lung, they localize there and they can produce IL-17 that recruits neutrophils that go to the lung and perform their job. They do phagocytosis, they release the molecules that they have. In particular, they can release myeloperoxidase and they can release elastase, and we have a paper submitted in this moment about this phenomenon. And IL-17 is able to increase the capability to provoke damages to tissue as well as to prolong their survival. And what was interesting for us is that after the first observations that we made, other groups all over the world confirmed what we have done. And indeed, this is a very nice study performed in 2 different locations, in Atlanta and in Hong Kong. And what came out is that studying 40 samples from patients with pneumonia in Atlanta or 100 sample patients from Hong Kong, it came out that the same populations that we found altered were found altered also in this study with 2 cohorts. And interestingly, they also found a consistent change in the amount of not only of T cells, of CD8 T cells, but also plasmablasts and plasmacytoid dendritic cells. And I will show you in a moment other data regarding plasmablasts. Then other papers came out, and this is another very nice paper that was able to analyze T cell population from the acute phase to the recovery. And the authors of the paper, the group of Rodriguez, found that the proportion of different cells, both CD4 and CD8, subpopulation of CD4 and CD8, were changing consistently during the infection. And doing a very sophisticated approach, this group of scientists found that a number of T cell populations followed different trajectories for the recovery after the acute phase of the disease. And in particular, you can see here how the trajectories of naive cells are different from those of memory cells, from those of CD16 positive T lymphocytes or CD57 positive CD8 or CD4 T lymphocytes. So to make a long story short, the story was, we know that we have consistent changes in T cells, but the question at that point was, nice, but what happens if you consider the T cells that are specific for the virus? Well, one of the first studies showed that there is a robust T cell immunity, and this immunity with cells able to produce interferon gamma after being activated with viral peptides. These cells are present during the infection, and they are very strong, and they do respond to different parts of the virus, such as the spike or the M protein, and the response is present immediately in the primary infection. But what was interesting for us was to understand one very important thing. I mean, we know that cells are able to produce interferon, but we can have other molecules than interferon that are produced by T cells, both CD4 and CD8. And in particular, what comes out in our hands was that we tried to study the polyfunctionality of SARS-CoV-2 specific CD4 and CD8 cells. And what came out is that if you cope CD4 cells with different peptides from the virus or from a controlled virus, which is the SARS-1, the old one, you can find still a response to different peptides. And what is important for us is that there is a very complex polyfunctional assay and polyfunctional trait, in different patients. So healthy donors are different from patients with COVID, from those who had a pouch symptomatic COVID and recovered, or from those who had a serious COVID and had recovered. So the same phenomenon was present among CD4 as well as among CD8. So the polyfunctionality, in our opinion, is something that we have to consider, especially now, because we need more sophisticated assays to understand how the immune response to COVID works in general population, not only in the study groups during clinical trials. So I think that we have to be very careful in saying that there is no response from patients who had a low number of antibodies, because very likely they can have a very strong T-cell response. And what was found also is that the T-cell response can be different from those against other viruses. In this study, it was shown that the SARS-CoV-2 spike-specific CD4 or CD8 T cells are different and not phenotypically identical to cells that are specific for other viruses like cytomegalovirus. So in other words, if you study in the same sample cells that are specific for one virus or for the other, and you stain with a number of other different markers to see the phenotypic or functional characteristics of the two populations, those that recognize SARS or those that recognize cytomegalovirus, as you can see here, they migrate in different parts of the graphic, so it means that all the other phenotypic or functional characteristics are different. The good news is that they work well and they can be studied in vitro with some little technical problems. Then the question was, how long does the immunity last? And in this very first paper by the group of Breton in Journal of Medicine, it was shown that the T-cell immunity in patients who recovered from COVID-19 can be as long as 6.1 months, simply because the study was interrupted at that time. So it is very likely that we can find even now T cells against the virus, specific T cells, even after a much longer period. And another study was, in my opinion, very important for us, and it's the study by the group of Alessandro Sette. And what they found was that if you study different types of T-cell memory and B-cell memory, you see that you can have a response to, after 6 months, you can find specific responses to the virus among CD4, CD8, B cells, different type of IgG or IgA, and you can divide the response in 5 different categories. And in after 6 months from the infection, you can find that a number of people, those in black or in red, can have more than 4 different responses to the virus. So it means that the immunity against SARS-CoV-2 has a long duration and can be really important for the health of the patient. Then we took a look as well to the specific T-cell memory, and in collaboration with a group of Pia Kvisborg in the Netherlands, we studied a number of patients during the acute phase. Actually, most of them were in ICU. We studied 31 patients and we coped them with about 500 peptide class 1 complexes that were restricted to 10 common HLA alleles for Italian people. So we identified 18 CD8 T cells that recognize epitopes that included an epitope with immunodominant features that derived from the ORF1AB and was restricted to HLA A0101. So in characterized that population of specific T cells, and we found that these cells had, at the moment of the infection, markers of exhaustion. They lacked cytokine production and had a gene profile characteristic of cells that were undergoing functional exhaustion. But after 6 months, after 5 months actually, if you repeat the analysis in the same patients, you can see that the markers of exhaustion are disappeared and cells start working again. Then here is the gene trait of the cells that I mentioned to you, and after 6 months, markers of activation appeared. So what happens in other situations? Well, we know that the infection of pregnant women is very important for several reasons. So we know that the immune system of pregnant women is characterized by an anti-inflammatory trait, and for this reason, several immune disorders can undergo a sort of remission. And in theory, the point is that we have it shifted towards a Th2 immunity, as I mentioned, could leave the mother, however, the shift can leave the mother more susceptible to viral infection because Th1 response can better help to contrast viruses. So we have a number of questions to answer, such as why symptom and severity of the disease apparently are different in most pregnant women, not all of them, but most, and if the infection can cause complications, and if we have a transmission of the virus to the baby. So if we have a vertical transmission that we have to consider. On the other side of the coin, we have aging. Aging is means for us inflammaging, so the immune system is driven towards a Th1 direction, and all the individuals had a number of markers which are very characteristic of the aging process, such as restriction of the T cell repertoire due to the thymic exhaustion, alteration between naive and memory cells, changes in B lymphocytes, diminished lymph monocyte functionality and capability to present antigen, and so on. As a result, old people are more prone to develop a number of infections and to diminish the defenses against cancer. And we know very well from the very first moment that the impact of SARS-CoV-2 in old individuals was absolutely dramatic in terms of morbidity and mortality, and this became one of the most important problems of the pandemic. So we, in collaboration with the group of Fluidigm, we had the possibility to perform a number of studies. First of all, we could use 38 parameters flow cytometry to investigate very carefully all possible PBMC population, paying a particular attention to TB and B cell compartments. Then, in collaboration with Biotechne, we could measure the plasma level of 62 molecules, cytokine, chemokine, and so on. And then we studied the production of cytokine as well as the T cell and B cell proliferation capability and the expression of mast regulator genes. So this is a paper that is under revision now in a nice journal showing the population of pregnant women that we have studied. All of them had asymptomatic or post-symptomatic infection with a low fever below 37.5 degrees Celsius, a little cough, a loss of smell and taste, but in some cases, half of the cases, they had absolutely no symptoms, and they were found positive when they came to the hospital for a routine control. We characterized the PBMC by 38 parameter mass cytometry with the lanthanides, and here on the left you can see the target, the antigens, the clones of antibody used, and the tag, and it was very simple to identify a number of different populations. The main are here: CD4, CD8, mite cells, monocyte, dendritic cells, NK and B cells, and also low-density neutrophils that, if you perform a classical cycle, they migrate into the area of PBMC. So they are not actually lymphocyte or monocyte, but they migrate in the same area. What happens? When we compared all the possible populations that we found, and this is only a part of the populations, in pregnant negative women against positive women, positive means infected women with SARS-CoV-2, what came out is that there was practically no difference in the amount of all populations you can imagine. The only difference in between SARS-CoV-2 negative and positive was here indicated by the green arrow, and was related to the presence of a significant amount of low density neutrophils among the lymphocyte and monocyte populations. Then we perform a more detailed characterization of CD4 cells and CD8 cells, and also in this case we found practically nothing no changes among CD4 cells, no difference between pregnant positive or pregnant negative. The same phenomenon was observed for CD8 cells. We found some differences between between non-pregnant women of the same age and pregnant women, both positive or negative for SARS, and in small changes in CD8 cells, very few changes in B lymphocytes as well. The expression of mast regulator genes and chemokine receptor gave the same results. We found very similar data showing very similar numbers and percentages of cells expressing T-beta, γ3, CSR6, and so on among women not pregnant, or pregnant with or without SARS-CoV-2 infection. Then another study was related to the T-cell proliferation and T and B cell proliferation. And again, to make a long story short, what came out is that no changes in T-cell proliferation among women with or without SARS-CoV-2 infection. The question came out, what about cytokines? We know that cytokine storm is a dramatic event in patients with SARS-CoV-2 infection, and in fact, we show in April 2020 the amount of the cytokine storm in patients with COVID, and we show that a number of cytokines, both inflammatory or anti-inflammatory, can change. But the most important thing that we found was that if you are able to inhibit the production or the activity of some inflammatory molecules like interleukin-6, you can really save a number of patients. And I am very proud to have published this study in collaboration with a group of infectious diseases in Modena, Cristina Mussini, Giovanni Guaraldi, and others, and Massimo Girardis from the ICU unit. And we found that in a retrospective study published in August 2020, if you treat patients with tocilizumab, which is a drug able to block interleukin-6, you can have a consistent and dramatic reduction in the number of patients undergoing mechanical ventilation or death. And this was a retrospective study that was confirmed almost 6 or 8 months later by a randomized study published in the New England Journal of Medicine, and now by a second study, a randomized study published in the Lancet. So in other words, if you block inflammation, you can save patients. So the question was, we know this, but what happens in pregnant women? Well, I don't want to be boring, but also in the case of the analysis of soluble molecules, we found very little differences between pregnant women with or without SARS-CoV-2 infection. What we found, however, was an expression which was quite interestingly increased of molecules like interleukin-1 receptor antagonist, while we found a decrease in molecules like IL-27 or PD-L1. So the impression that we had was that in pregnant women with SARS, there was a small increase in the amount of molecules able to contrast inflammation, anti-inflammatory molecules, in comparison with those that provoke inflammation. Then we perform a very complicated story with a multi-correlation analysis of all parameters, and interestingly, we focus our attention of low-density neutrophils. You see the population on the left, there is a green bar, maybe it's too little, but the story is that the amount of low-density neutrophils was very much correlated with a number of molecules like April, IL-13, IL-17E, CSL7, IL-18, BAF, CD4 ligand, interferon gamma, and so on. So what we found was that, to make a long story short, there is an increased number of neutrophils, or low-density neutrophils in blood, but this increase is accompanied by an increase of molecules that are able to block inflammation, likely induced by these cells. Finally, the last thing that we checked was the production of cytokines in vitro, and we checked the production of a number of cytokines, as we have done in the studies before. We checked IL-4, IL-17, TNF, and interferon gamma, and among CD4 or CD8, and we found no changes in the production of such cytokines in CD4 or among CD8. Then we directed our attention to aged people. Aged people, because, you know, aging is the other face of the medal. We studied the SARS-CoV-2 infection in the context of the aging process, in which you have a number of alterations and you have a number of changes in that regard. The mitochondria, you have loss of proteostasis, autophagy, you have changes in apoptosis, you have an increase in cell senescence, you have immune senescence, inflammation, inflammaging, and alterations in the microbiota, changes in the endocrine system, and so on. And so we put in this cartoon that was published in AIDS concerning patients with HIV infection, we put the therapy for the SARS-CoV-2 infection, and we put the SARS-CoV-2 as such. In fact, we think that SARS-CoV-2 and its therapy can enter into the very complex situation that you have in aging persons. And what have we done? We studied a number of people with COVID-19 pneumonia, a total of about 60 patients. 35 were below 60 years of age, and the mean age was about 55, and 24 patients were over 70 years of age. And they were compared with a total of 67 healthy donors of different age from 27 to 101 year. So what we have done, same as before, in collaboration with IGME, we have performed a 38-parameter analysis of PBMC, and we were able to identify a number of different population like CD4, CD8, and K, and so on, and also a consistent number of low-density neutrophils. But then we focus our attention on the differences between patients below 60 years against patients over 70 years of age. And what came out is that a number of populations were different among these two groups, and in particular, we found a significant change in CD4 central memory cells, in CD8 naive cells, in memory B cells, in plasma blasts that were increased in people over 70 years, and in low-density neutrophils that apparently are a very important population to consider studying changes due to COVID-19. We then performed an unsupervised analysis using the principal component analysis, and one of my collaborators, Domenico Tartaro, was very clever. He was able to identify, by using an unsupervised assay like principal component, that you can cluster. If you put all the parameters that we have studied together, you can cluster patients on the basis of their age. So you see here on the left, patient over 70 years, they are in the PCA graphic on the top, in the upper left quadrant, while patients below 60 years of age were in another part of the graphic, on the bottom left part. And the most important parameters that contributed to this phenomenon were, in some cases, plasma blasts, as well as naive CD4 cells or memory B cells for healthy individuals. So, in other words, the differences were so strong that you can find them also using unsupervised method of classification. Concerning T-cell population, what came out is that patients over 70 years had a low T-cell proliferation among central memory CD4 cells. The same phenomenon, a low proliferation, was observed among CD8 naive T cells, while an increase in cell proliferation was found among B cells, very likely because of the presence of a significantly higher number of plasma blasts. Then we took a look to the cytokine storm, and again, with a panel of 62 cytokines, we found a number of molecules that were differently present in plasma of patients under 60 or over 70. And what is most striking for me is the presence of a significantly high amount of interleukin 6 in the plasma of all the patients. So it means that very likely the inflammation process and the production of inflammatory cytokine like IL-6 is the driving force of all the changes that we saw in old people with SARS-CoV-2. And of course, we found a number of other alterations in different population, in different cytokines or plasma factor, and if we could speculate, for example, that the amount of soluble Fas and soluble Fas ligand could be important to Fas soluble is a marker of activation, while Fas ligand is a marker of control of activation. So we found more Fas and less Fas ligand in all the individuals. But interestingly enough, we also found a number of other changes in, for example, in chemokines like CCL2, CCL2, CH3, CL1, and so on, but also in molecules like leptin, like endothelial growth factor, and others. So, in conclusion, in our cohort of pregnant women, SARS-CoV-2 had no impact on newborns. No newborn was infected or suffered of any disturbance after birth. So we can hypothesize that infection of the fetus does not occur because of a reduced co-expression and co-localization of placental ACE2 and transmembrane semiprotease 2, and that the typical immune suppression that we see during pregnancy, or I would say the change in the immune response during pregnancy, could protect not only women from the cytokine storm, from the massive immune activation and hyperinflammation, but could also protect the fetus from a possible maternal immune aggression. And this data might be important for clinicians because if the condition of the patient are stable and the infection remains as mild or asymptomatic, there are no risks from pregnancy, and there are no complications neither for the mother or for the newborn. So that in a large part of the cases, an anticipated delivery should not be required, and this is something which is clinically relevant. As far as the aging process is concerned, we have shown that people who have an immune system skewed towards stage 1 because of the aging process and because of the inflammation can have an enhancement of the production of inflammatory molecules and can undergo serious damages. And we know quite well the phenomenon, and we are trying now to understand which is the role of the innate immune system. These data that I show you are not published, so I'm very happy to show to the community of immunology people all over the world what we have done. So maybe some of you could do much better work, and I would like to see better work than ours. And in any case, what we can say is that having learned very well the immunopathogenic mechanism that the virus has and the virus uses to fight the host, could allow us to develop new therapeutic strategies able to hit in multiple targets, especially in old individuals in which the vaccinations are likely less effective than in young people. So all over the world, the technologies allowed us to develop a number of vaccines that are, of course, activate T cells and B cells, and they are saving countless lives. However, the question is how vaccines work in different groups of people that we have to consider, including those of advanced age. And finally, I want to acknowledge a number of people who we are working with since almost 1 year and a half, the group of infectious diseases in University of Modena and Reggio Emilia, especially Cristina Mussini, Giovanni Guaraldi, and all the others, the group in the ICU, Massimo Girardis and Marco Sita, the group in the respiratory disease unit, the laboratory medicine, Pia Quisborg, Tom Schumacher, and Anastasia Ganjev in the Netherlands, in Amsterdam, and the group of fluid dyeing led by Andrew Kwong, who helped us in performing a number of analysis of PBC with the 38 panel that was developed for this reason. And I also want to thank Mariella Zuccalai and her group for hosting me here. I am not in Modena, I'm in another city, and they were very kind to give me their office to present my data. And finally, the last slide is showing the eyes of my group with Lara and Sarah, who are the main forces of the lab. Sarah for the flow cytometry and Lara for the part of molecular biology, including, and then the group includes Rebecca, Anita, Caterina, Alicia, Lucia, Anna Maria, Domenico, who is the bioinformatics guy with Marco Mattioli, Anna Maria Paolini, Laura Franceschini, and finally, of course, I want to thank you for your attention.
Well, thank you very much, Andrea, for your outstanding contributions and also for such a remarkable lecture. We have enjoyed every piece of work you have presented. Now we have some questions, many, I would say. And we will do our best to cover as many as possible. Okay. In inflammaging, the Th1 immune response profile is predominant. If in COVID-19 there's an increase of IL-17, does it make changes in these immune profile preferences in elder patients, or they still have a Th1 direction immune response?
Now they still have a Th1 direction, and what we found is that all the markers of inflammaging, including the decrease in naive cells, an increase in memory cells, an increase of activation was present in all people with COVID. So our impression is that there is a sort of boosting of inflammaging by the response to the virus. And this is one of the reasons people in comparison with young people.
Okay, thank you. We have here another question. Is there any involvement of SARS-CoV-2 in the bone marrow? What is the implication of the high frequency of low density neutrophils?
Well, there are no evidences of this. My impression is that the release of low density neutrophils can be the increase could be due to two different phenomena. One could be the release of immature cells produced by the bone marrow because there is a very high amount of GM-CSF produced during the COVID infection. So GM-CSF can induce the production of new neutrophils that are released by the bone marrow when they are immature. This is one hypothesis. The second hypothesis, which is not in contrast with the other, is that low-density granulocytes are those who had already released the granules. And so they have done their job, and they are old cells, in a way, that had already produced molecules like myeloperoxidase and elastase, which are able to induce serious damages to the host. So two faces of the same coin. GM-CSF can induce the release of young, very young and immature cells. On the other side, IL-17 can keep cells alive, so even after degranulation, neutrophils can be present in the periphery.
Great, thank you. From Shuba, what are the MDSCs, the myeloid-derived cells, levels in pregnancy and in aging during COVID infection?
I don't think that this was studied enough. So we missed data on a number of cells, including the myelosuppressed debris cells. We have still to study this phenomenon. Thank you for the very important question, and I will take the suggestion. Thank you to our friends in Cuba.
How do you think the variants of SARS-CoV-2 might influence the immune response? Could you give us your perspectives now?
Well, the $2 million questions. I mean, of course, the variants are important, but in my opinion, I don't see how the variants that we have now, today, I mean, on July the 5th, could differ one from the other in terms of the immune response. In other words, I think that the changes that we see in the variant, I mean, the higher capacity to bind the ACE2 receptors because of mutation in spike are not so crucial to change the immune response. They are crucial because they are more infective, so they can infect more people. But if we take 100 persons who got the infection, I do not think that one who has one variant goes in a different way from one who has another variant. So in other words, I don't think that the variants could influence the immune system in a consistent way. But I'm talking about what happens on July the 5th and the day that we have today, because we learned with this bloody pandemic that what you say today can be wrong tomorrow. and maybe tomorrow we have, I hope not, but we could have a variant which is more aggressive, replicates in 10 minutes instead of 10 hours or whatever, and is much more devastating for the host. In this moment, this is the idea. Interestingly enough, we know that vaccination, if you cover people with the 2 doses of vaccine, either DNA vaccines or RNA vaccines, you are protected in a very good way, not only from the infection, which is, we have a good protection. The data today say that if you complete the cycle and you have 2 doses of DNA vaccine, like AstraZeneca, you have a protection of about 60% from the infection and more than 90% from the severe disease. So you could be infected, but you don't go bad. I mean, you're okay. On the other side, if you receive an RNA vaccine, 2 doses of the RNA vaccine, Moderna and Pfizer, which is those that we have in Italy and in Europe, I mean, what happens is that you are protected at about 70% from the infection and more than 95% from the severe disease. So it means that immune response works against the vaccine. The vaccine are based on the original virus, the one that arrived from Wuhan in January last year. And these vaccines, again, that are directed against the old virus, are able also to protect from the variants that we have now on July the 5th. I have no idea of what can happen tomorrow.
Okay, just lost the question. Oh, sorry. Can you this is related, absolutely. Can you tell us about the different cytokine reaction from the additions of the SARS-CoV-2 virus? Is there any important already discovered?
Well, I think that what is important, in my opinion, is that we have to consider that not only the single molecule, but the pool of pro-inflammatory or anti-inflammatory molecules. So in other words, we can have the balance. The balance is what counts. I remember the story of inflammaging. When we created the word inflammaging more than 20 years ago, the story was that we studied centenarian people over 100 years of age, and what came out is that they, in the plasma, they had a huge amount of inflammatory molecules, IL-6, TNF, and IL-1. So the idea was that aging is related to inflammation, and so inflammaging. But at that point, we were a little bit naive, and especially we did not know that there were other molecules like IL-1 receptor antagonists that you could measure, or IL-10, or whatever, anti-inflammatory molecules. So what came out later is that people over 100 years, they have a huge amount of inflammatory molecules, but also of anti-inflammatory molecules. So the balance between inflammation that you need to start an immune response and anti-inflammation that you need to dampen and to calm the immune response is likely crucial. So my impression and my suggestion would be not to study only inflammatory molecules, but also anti-inflammatory molecules.
Good. For our wrapping up, could you elaborate on the role of other comorbidities in elderly population with COVID-19 dying in higher rate?
Well, unfortunately, we know well that comorbidities are crucial, and those people who have comorbidities, which are not very uncommon during aging, especially in the old age, over 75 or 80 years, those who have comorbidity are the favorite target for the virus, because the virus killed a number of people with obesity, diabetes, cardiovascular disorders, pulmonary disorders, and so on. So the coupling aging as such with comorbidity is a mortal combination, and the virus is really strong in those people who have comorbidities, likely due to the consistent derangement of the immune system during these comorbidities, and to the different asset of the lipids, of the glucose, and so on, and finally to the type of cells that are responding in a different way to a stimulus because of the cell vaccinations.
Thank you. Unfortunately, our time is over. Thanks again for your time and your outstanding contributions. Our special thanks goes to Olga Bon, also Anna Claire from IOIS, Fluidine for your support, and of course, Immunopedia for providing a bridge to expand immunology without borders. Please stay safe and take care of your loved ones. Thanks. Thank you, Andrea.
Thank you.
See you in Cape Town, 2022.
I hope so. Perfect. Always a pleasure. Bye-bye.
Goodbye, colleagues, friends.